Okay, great. Good afternoon, everyone. Is my voice okay? Yeah. Okay, so I'm delighted to introduce our speaker today, Dr. Kunliang Guang. Dr. Guang is a distinguished professor in the Department of Pharmacology and the Morris Cancer Center at the University of California, San Diego. He received his bachelor degree of science from Hangzhou University in Zhejiang, China, and his PhD at Purdue University. He then did his postdoctoral research in Jack Dixon's lab, I know that's good, at Purdue University, where he made multiple discoveries, including the dual specificity protein phosphatase family. Early works from his own laboratory led to the cloning of human MEK, MEK1 and 2, and elucidation of the mechanism of MEK activation. Kunliang has received numerous awards and honors, including the MacArthur Fellowship and the ASDMB Young Investigator Award. In recent years, Kunliang's laboratory has been studying the mTOR and the Hippo YAP signaling pathways in biology and disease, including cancer. So today, the title of his talk is Hippo signaling and cancer. So there's finally the housekeeping thing. Please post your questions in a Q&A that tab, not in chat, Q&A. So I will read out questions at the end. So our seminar will stop by 5:05. If your question cannot be addressed during this the seminar talk, and Claudia will mail your question to Dr. Guang. So that's all. I think that's it. So, okay, great. The floor is yours, Kunliang.
Okay, so let me share my screen. Do you see my screen?
Yes.
Okay. Well, I really would like to thank Jean for the hosting my seminar. I'm delighted to give talk at the Premium Cancer Institute in the world. So my title will be Hippo signaling and cancer. There's one potential conflict of interest I will declare is I'm a co-founder of Vivace Therapeutics. Actually, we will use some of the inhibitors that developed in our research. Okay, so what I'm going to do is I'm mainly going to summarize some of the was an understanding of the Hippo pathway regulation for the first part of my talk. And second one, I will talk about two ongoing studies research in my lab. So the Hippo pathway initially was discovered in Drosophila and named after this gene called a Hippo. Actually, the very first gene in the Hippo pathway was identified as a gene is Warts. And the Hippo pathway is completely conserved in mammalian system, consistent, so the mammalian genes, so the colored out mammalian genes consist of two protein kinases, MST and then LATs, which function as a tumor suppressor generally. And these kinases inhibit the transcription module, includes a YAP-TAZ transcription coactivator, which binds to the TAD family, 4 of them in our genome, DNA binding protein, to turn on gene expression. And those genes involved in a very wide range of cellular functions and physiological regulations. So one of the hallmark of the Hippo phenotype is tissue overgrowth, like mutation in Warts results in tissue overgrowth. it controls organ size. So here's the inducible YAP expression reported by DJ Pan and Fernando Camoga's lab 14 years ago, that YAP induction results in large liver. And this liver organ size control is reversible. If we remove YAP induction, we'll get to the normal size. So the Hippo pathway is very important in organ size control. And at the same time, we also discovered that YAP protein is highly elevated in liver cancer. About half of the liver cancer has a high YAP protein level. Now, so regarding the function, actually the Hippo pathway is very important from development, tissue homeostasis, and dysregulation to cancer. So one of the very first cell specifications during early embryonic development. So the exterior cell of the extremely early embryo will differentiate to become trophoblast, which eventually becomes a placenta. So this is the very first cell differentiation during development. And YAP has a critical role to modulate the trophoblast differentiation. And, so this is work from Sasaki's lab, where showing you can see the exterior cells has a nuclear YAP, which is active, and the YAP activity is able to driving the expression of CDX2, which is a key determinant transcription factor for trophoblast, eventually, as I said, become placenta. So YAP activity is both necessary and sufficient to drive the trophoblast differentiation. Now, in adult, the Hippo pathway is important in wound healing and tissue regeneration. For example, early work from DJ Pan's lab showed if you knock out YAP in intestine, normally the animal is okay, YAP knockout. However, when you give drug such as DSS to cause intestine damage, the wild type animal is eventually able to recover and survive. But the YAP knockout animal die very rapidly. And the reason you can see the intestine regeneration and differentiation is completely messed up. So that's give you example how important of the Hippo pathway in tissue homeostasis. Regarding cancer, the TCGA completion of TCGA analyzed almost 10,000 samples showed many pathways are altered, the very well-known such as RTK-RAS, P53, and Hippo is one of the newest among the signaling pathway, which are commonly altered in human cancer. So that's also really provide evidence, genetic evidence supporting an important role of the Hippo pathway in human cancer. So regarding the regulation, how the Hippo pathway, so the very first observation we made, so this was done by a former student at Bing Zhou. So he observed that when cell at low density, YAP is nuclear, here's the YAP standing. When cell at high density, YAP becomes cytoplasmic. And most of the cells, such as fibroblast or epithelial cell, display that nuclear cytoplasmic regulation. And if you look at the protein stability, another thing we noticed that at the low density, YAP is pretty stable. At a high density, YAP is unstable. Sorry, it's covered part. It's okay. Yeah, okay. So being figure out the mechanism of this regulation, which essentially is the main, dogma for the Hippo pathway. So what we discovered is that LATs dependent YAP phosphorylation results in 14-3-3 binding. That is critical for YAP cytoplasmic localization. Additional phosphorylation results in YAP ubiquitination and degradation, also explains the low stability of YAP at a high density. And also Fernando Camoga, we discovered that cell-cell contact is the signal. Both both tight junction and adherence junction mediated by angiomotin as well as alpha-actinin, respectively, to activate the hippokinase cascade. So at high density, the hippokinase is active, YAP is phosphorylated, become cytoplasmic, the transcription program is turned off. And we also showed that YAP activity regulation is a critically important contributor to contact inhibition. So high YAP activity promotes cell growth, and the cell contact, high density, cell growth are stopped. In larger part is mediated by the YAP inactivation. Conversely, at the low density, the kinase activity is low, YAP is nuclear, and turn on proliferation program. Another thing we found is by Faxing Yu, he discovered that the serum regulated the Hippo pathway. So here we look at the YAP phosphorylation, here based on the mobility shift. So the phosphorylated YAP will migrate slow, the unphosphorylated migrate fast. So if you remove serum, the YAP phosphorylation dramatically and quantitatively shifted from unphosphorylated form to fully phosphorylated form. We mapped actually as a 5 phosphorylation site in the YAP. And Faxing purified from the serum and identified the lysophosphoric acid and a related lipid called S-sphingosine phosphate. So LPA and S1P are really potent active ingredient in the serum to induce YAP dephosphorylation and activation. So here's detected YAP phosphorylation by phospho antibody or by mobility shift. So as low as 0.1 micromolar LPA is able to induce full YAP dephosphorylation. And Faxing discovered that LPA actually acts through the G protein coupled receptor, or this part is not from the literature. And we found it is a G1213 coupled LPA mediates that signal downstream to the low GTPase, results in inhibition of the hippokinase cascade, therefore YAP dephosphorylation and activation. So in addition to that, recently, a year ago, we published that we also found many stress signals can regulate the Hippo pathway. And last year, we showed that heat shock is rapidly able to activate YAP. Here, example, we shift the cell from 37 to 43 degrees, and we see very rapid, within 30 minutes, completely YAP dephosphorylation by the phosphatase or the YAP phospho antibody. And the 37 degree also results in YAP nuclear accumulation. So high density cells, YAP is cytoplasmic at 0 minutes, and 30 minutes, it's become nuclear. I should say, we did not propose a set to 43 degree. Actually, we thought our incubator was 42. We did all the experiment, and at the end, we put a thermometer in, and turned out it's 43 degrees. So that's why it's kind of funny, 43 degree. And the heat shock induced YAP dephosphorylation is very rapidly. If we shift it to 37 degrees for 15 minutes, see everything come back. Lower temperature does not have that effect. And Ming found the mechanism. So heat shock is apparently different from many of the other signals we know, which act through MST MAP4K, which are the kinase phosphorylase. Heat shock actually appears to, at least in part, directly inactivate LATs. Mechanistically, is because heat shock protein, recruiting protein phosphatase PP2A, results in YAP dephosphorylation and inactivation. So and consistent with this mechanism, heat shock signal is dominant to all upstream signals. So heat shock signals are always dominant regarding LATs inactivation and YAP activation. So I would like briefly to summarize what we know about the Hippo pathway. So I started with the cell-cell contact signal. I mentioned about the hormonal signal, such as the G protein coupled receptor signal, regulates the Hippo pathway. Actually, I want to expand a little on that. G protein coupled receptor depends on which hormone you use. It can either activating or inhibiting the hippokinase, such as LPA inhibiting the hippokinase, while things like a glucagon, epinephrine activating the hippokinase. So it depends on which G protein is involved. In addition, I also mentioned about heat shock, where we previously reported that cellular energy stress, osmotic stress, all are able to regulate the Hippo pathway, actually activate the kinase. And there's also really interesting is the Hippo pathway is able to respond to mechanical signals, such as metric stiffness. And we have previously showed that integrin may be mediated, partly mediated mechanical signal to regulate the Hippo pathway. So we know a lot about the signal about the Hippo pathway upstream. I should say that's different from like a Wnt pathway or TGF beta pathway. There's no designated hormones or receptors specifically for the Hippo pathway. Yet the Hippo pathway is able to respond to many, many different signals. Now, one of the key questions is how MST, MAP4K, these kinase activity are regulated. Now we know MST, MAP4K needs phosphorylation to be active. We also know, actually, early work from your colleague Joe Everick's lab, he showed that those kinases probably autophosphorylate. They form a dimer and autophosphorylate and activate. So the key question is how upstream signal regulate MST MAP4K activity to regulate the Hippo signaling. So in order to understand that, I needed to introduce an interesting new component called a STRAPAC. STRAPAC stands for striatin, which is a protein, associated protein phosphatase and a kinase. Notice MAP4K is there. This was defined by Anne Cloud Gingras when she was using protein phosphatase subunit looking for interacting protein. So this is PP2A, scaffold protein subunit A, catalytic subunit C, looking for interact protein. She defined these very complex yet stable complex called a STRAPAC. And quite a few years ago, work from Nick Tapan's lab showed that the the STRAPAC maybe regulate Hippo activity. And generally the field thought this might be a negative feedback. The phosphatase, deactivate that. So we Ray-Chen, a postdoc in my lab, look at the interaction of MST with STRAPAC and the MAP4K with STRAPAC, and that's what the model would have, the topology based on our various co-IP. So then we want to look at the role of STRAPAC. So we deleted the striatins. There's two striatin genes, striatin 1 and a strip. Sorry, we deleted the strip. If we did the striatin, see the same results. We strip 1 and 2. And what we found is in the wild type cell, MAP4K activity is inhibited by LPA. We know that previously. Now, in the STRIP1-2 double knockout, the basal MAP4K activity is highly elevated. And most importantly, MAP4K is no longer able to be inhibited by LPA. And if we look at the other component, here's a wild type, here's a STRIP1-2 double knockout cell. So in the wild type cells, LPA start from serum stop the condition. If we add LPA, results in very dramatic YAP dephosphorylation. The STRIP1-2 knockout is completely gone. If we got the LATS kinase activity and phosphorylation, can see LATS phosphorylation dramatically inhibited, STRIP1-2 knockout is gone. And this one effect is specific, at least to the LATS and YAP. If we look at the MAP kinase ERK activation, stimulation is not affected by the STRIP1-2 knockout. So a lot of experiment, Ray come up with the following model. So based on Ray's data, we think in the absence of serum, under serum starvation condition, the phosphatase, PP2A catalytic subunit, is not associated with the STRAPAC, which is associated with the MAP4K. So therefore, MAP4K probably autophosphate is active, and the same model is true for MST, to phosphate the LATs and then YAP results YAP inhibition. Now, if we stimulate with serum or LPA, the first thing what happens is a rho GTPase is activated, which is downstream of the GPCR. And when rho is activated, rho binds to an effect called rho-filling, rho binds to rho-filling. Now, rho-filling is dissociated from NF2. NF2 is a very well-known tumor suppressor involving Hippo pathway activation, which results in association of the phosphatase together with the MAP4K, and therefore results in MAP4K dephosphorylation and inhibition. So we would like to propose in the Hippo pathway kinase cascade, it is the phosphatase which mediate upstream signal to modulating the kinase cascade. And then actually we also extended that true to many of the MAP4K family as well as MST. So this, I think, we were very happy. I think we found one of the mechanisms how upstream signal regulates the Hippo pathway. Now, if we read the literature in the Hippo field, basically most of the study is focused on YAP regulation. So the dogma for Hippo pathway regulation is phosphorylation dependent nuclear cytoplasmic shuttling of YAP. And that is a dogma. Now, YAP is a coactivator. Itself cannot bind to DNA. So TAD is essential, is required for YAP to bind to DNA to turn on target genes. So when, curious questions, does TAD, is TAD regulated by any signal? So Kim Lin, a former student in my lab, she looked at the endogenous TAD YAP localization in response to many different signals. And she found, as what some of them has been previously reported, that many signal-induced YAP cytoplasmic localization had a little effect on TAD. Until Kim looked for additional conditions, such as osmotic stress, or very high cell density, or cell detachment. Those are also able to induce TAD translocation to cytoplasm. So that was quite interesting. And Kim focused on the osmotic signal, and she found that actually the p38 MAP kinase, which is a stress-activated MAP kinase, responds to environmental stress to regulate TAD nuclear cytoplasmic shuttling. So therefore, now we know TAD also can be regulated. Now, in order for the transcription program to be turned on, the cell needs both TAD and YAP in the nucleus. And we know the Hippo pathway regulates YAP, does not directly regulate p38. p38 does not directly regulate YAP. So that's really provide a very nice mechanism of different signal integration at the transcription module to regulate gene expression, cell growth, or stress response. Now, TAD is a really interesting transcription factor. Work from Xiu's lab as well as Christian Matero's lab. So that showed that TAD. So here's a co-crystal structure of that. So the magenta color is YAP. The teal color is TAD. So in the TAD, there is TAD goes through palmitoylation. Now, the unique thing about the TAD palmitoylation is different from other palmitylation too. First, TAD palmitoylation is autocatalytic. If you have a purified TAD from bacteria and you give a palmitoyl-CoA, it will be able to palmitoyl itself. Second, the TAD palmitylation does not serve to stick TAD to membrane. A lot of palmitoylations, such as RAS, those kind of OSOX, those palmitylation results protein membrane association. TAD is not. the palmitate stay inside of the protein TAD. And it is a function. It's a function that is important for TAD. So this is really exciting because it provides a mechanism to targeting this transcription program, means by inhibiting TAD, looking for compound can inhibit TAD palmitylation. So Tracy Tan at Vivace Therapeutics, so we collaborate with them, we screen library, then go through a structure function study, eventually develop a potent TAD inhibitor in this VT103. Now, if we look at the TAD palmitylation, so here's a no label, no labeled palmitate, and we don't see anything. And DMSO, TAD very nicely palmitated. And the VT103, we have several of them, are very potently inhibit TAD palmitylation, very effective. And interestingly, when we look at the tumor growth, so this cell line has a TAD deletion, not TAD, has NF2 deletion. NF2 is upstream activator of the Hippo kinase. So when NF2 is deleted, YAP is dephosphorylated and active all the time. So we can see this cell line, we use an inhibitor, increasing concentration inhibitor. And when the tumor is very small, about 1 minute, just barely palpable, when we add the drug, and you can see it's very potent to inhibit tumor growth in xenograft model. And in vitro, we demonstrate that those tumor cell lines, the left 3, has a mutation or deficiency in TAD, in NF2. NF2 is a tumor suppressor, neuron fibrinogen 2. They are sensitive to the TAD inhibitor. Those tumor cell lines has wild type TAD are not sensitive to the TAD inhibitor. So this clearly suggests some of the specificity. And currently, the compound is in clinical trial to test them for cancer treatment. So one of the function question always puzzled us is, what are the key target genes in mediate downstream YAP to mediate organ size control, right? So if you have a wild type, if we induce YAP in liver, the liver become very big, and this is reversible. It's very fast, only about like a few weeks, couple of weeks. So Jay, as a postdoc in lab, he found that actually AP1, more specifically X, the Jung family proteins, as the fourth family proteins, are downstream of YAP to mediate the organ size control. So here I show you one data. So AP1 is a transcription factor heterodimer, Jung fourth family, and a T5224, which is an inhibitor, AP1 inhibitor. So the wild type animal, if we treat it with induced YAP, the liver getting very big. Now, if we treat it with the AP1 inhibitor together with YAP induction, you can see the liver increase of liver size is completely blocked by AP1 inhibitor. So what we found is, among the YAP target genes, fourth family are induced, which results in increase of AP1. And we propose that YAP alone or YAP teed together with AP1. Actually, about more than half of the TAD binding side also has AP1 next to that, induce the genes. So those genes collectively contribute to the organ size. So we conclude that AP1 is a key component of downstream of YAP involved in organ size control. So for the rest of the time, I'm going to talk about 2 ongoing projects. Our study to study the Hippo pathway, and include one of the really surprising results, which I'm going to spend most of the time on that, is the functional interplay between Hippo and estrogen receptor. Now, so my postdoc fellow Shen Hong Ma, who deleted LATS1 and 2 in breast cancer. Actually, he first deleted in the MCF7 cell, breast cancer cell line. It was very surprising. He found that, so he has a two independent deletion. He found the knockout cell, LATS knockout cell, MCF7 cell grow much slower. And that was very surprised because the LATS 1 or 2 generally are considered as a tumor suppressor. So while we delete the tumor suppressor, the cancer cell grows slower. So he made an additional deletion and he found the following. He found that the ER positive breast cancer, if we delete LATS, the tumor cell grows significantly slower. The ER negative breast cancer cells, if we delete the LATS, do not have much effect. So apparently those are LATS independent, those are more like LATS dependent. So we really want to know, how could a deletion of tumor suppressor results in inhibition of cell growth? So we did RNA-seq, and we found that, as expected, the LATS knockout dramatically increased the YAP signature. What we found is very surprising is that the estrogen receptor transcriptome, the ER responsive transcriptome, is almost completely abolished in the LATS knockout cell. So that suggests LATS is required for ER-dependent transcriptome. And when we look at the gene expression, it becomes very obvious. So we found that estrogen receptor ER alpha coding gene, ESR1, is almost completely blunted in the LATS 1-2 double knockout cell. So here's the mRNA, and if you look at the protein, estrogen receptor protein is completely gone. This LATS knockout, and it obviously, as predicted, YAP phosphorylation is gone. So here's the YAP protein. We also confirmed that in a primary breast tumor organoid, and we, in fact, it was LATS CRISPR, and you can see, along with LATS 1-2 knockout, ER level is decreased. We look at it in organoid culture using mouse cells. So we have the conditional. So the LATS 1-2 flox mice. We infected with Cree, not mice, tumor tissue organ, normal tissue, mammary tissue organoid. If we infect the Cree to delete the LATS 1 and 2, and the estrogen receptor is gone. We also look at a transgenic animal overexpress TAS. TAS is a YAP homolog, function partially redundant. You can see here the TAS expression, and in the wild type cell, there's a estrogen receptor expression in the epithelial cell, and in luminal epithelial cell, you can see ER for staining is significantly reduced in transgenic animal. So that's the results demonstrate that LATS are really important to maintain ER alpha expression. We look at whether that is mediated by a YAP test, and the results is definitively yes. So in the wild type of cells, LATS knockout abolish the estrogen receptor. In the YAP test double knockout cell, first the basal level increased slightly, and most importantly, now co-deletion of LATS cannot suppress ER alpha expression. So YAP tests are required. Actually, YAP tests are also sufficient. Overexpressor of a YAP, active YAP, repressed endogenous ER alpha expression. This TAD binding defective mutant cannot repress that. So that means let's maintain ER alpha expression by inhibiting YAP tests and dependent transcription. Now, YAP tests are transcription coactivate, at least in our hands. Most time it stimulates transcription. So how YAP tests repress ER expression? So we propose it might be indirect, means some of the YAP test target gene may mediate that effect. So we look at the YAP test, we look at the gene expression between wild type and LATS 1-2 knockout cell in several breast cancer cell lines, as well as tumor and tissue organoid, because we know that LATS dependent ER regulation is coming in all ER positive cells. We look at that, and then we're looking for what genes are commonly regulated. And what's really interesting to us, caught our attention, is VGL family. So in this case, we compare the T47D as well as MC7 mRNA, which is commonly induced. VGL3 and VGL1 are strongly induced. So reasons those are really interesting because previously it is known VGL4 is able to bind to TAD and mutually exclusive with YAP, means VGL4 may able to kick out YAP and to repress YAP target genes. We also notice in the mammary organoid, VGL3 is strongly induced, as well as VGL1. So we tested about overexpression, really surprising. Expression of VGL2 or 3 is sufficient to repress ER alpha expression. So actually VGL2, 3 are more homologous to each other, and they are more distantly related to VGL1 and VGL4. And now here's the mRNA level. So our data suggests VGL2 and 3 can repress ER alpha. We focus on VGL3 because only VGL3 is expressed in breast cancer cell and tube organoid upon YAP activation. So we look at the function of VGL3 in ER alpha regulation in response to Hippo deletion. So in let's knockout cell, ER alpha is gone. So we first deleted VGL3. There's a slight increase, not a dramatic increase of ER alpha. What is important is when VGL3 is deleted, co-deletion of LATS1 and 2 are cannot abolish ER expression. Also still decrease slightly, but you can see it is not able to completely do that. And here's the quantification at the mRNA level. So this data suggests that the VGL3 mediate part of the Hippo signal to repress estrogen receptor. And so we tested whether the VGL3 act through the TAD. Of course, we confirmed the VGL3 binding to TAD, and the results is very clear. So in the wild type cell, overexpression of VGL3, yes, VGL3 overexpression results in ER alpha repression. If we delete the TAD, so delete all 4 TAD, the TAD is gone. Now we overexpress VGL3, the ER alpha is not affected at all. That means VGL3 depends on TAD to repress ER alpha. If we do the same experiment with YAP TAZ double knockout, VGL3 still can repress ER alpha. Now, we also look at the genomic binding. We performed ChIP-seq, chromatin immunoprecipitation, and DNA sequencing, and we found that the VGL3 peak and the TAD peak is almost completely overlapped. Furthermore, in the VGL knockout cells, you can see very little in the TAD knockout cell, very little VGL3 binding to DNA. So from that, we propose this model. VGL3 bind to TAD to repress ER alpha. Actually, we found there's multiple TAD binding sites in the ER alpha super enhancer. So the next question we want to ask is mechanistically, how VGL3 function as a transcription repressor? I should say virtually little, there's almost nothing has been done published about VGL3. So in order to do that, we performed a protein interaction, looking for what is VGL3 interacting protein. And here are the lists, the top VGL3 interacting protein based on the IP and mass spec. Those are implicated in transcription. And we found many are involved in transcription repression. And of course, we also found the TAD, so that's very nice, assuming the IP is working very well. So we knock out each one of them, then look at which knockout can block the effect of VGL3 overexpression or LATS1-2 double knockout. And we only found this one, NCOR1. This is a very well-known co-repressor. When NCOR1 is knocked out, VGL3 overexpression cannot repress ER alpha, at least cannot efficiently repress ER alpha. When NCOR1 is knocked out, LATS knockout cannot repress ER alpha. So our data suggests that VGL3 recruits the repressor to the TAD binding site in the estrogen receptor gene super enhancer to repress its expression. So finally, we want to look at the clinical implication. So we found that LATS is required for the cell to respond to estrogen. So these cells, MCF7 cells, if we add E2, estrogen, cell growth is stimulated. In the LATS1-2 double knockout, this stimulation is largely compromised. And furthermore, if we xenograph the wild type LATS2 double knockout cell in the mouse, the tumor growth also strongly inhibited. So we want to test, this effect is mediated by estrogen receptor. It's a specific effect. So in the LATS1-2 double knockout, we re-express the ER alpha to the level similar to endogenous ER. This is very important. Actually, too much expression, the cell cannot tolerate. And now we look at, colony formations. ER alpha expression rescues the growth defect of the LATS1-2 double knockout. So again, we collaborated with Vivace, Tracy Tan, developed a LATS kinase inhibitor. So VT 02956 is a very potent sub-nanomolar LATS kinase inhibitor. A related homolog, which is structured very similar, but is completely inactive. You can see this is in vitro kinase assay, inhibit LATS1 and inhibit LATS2. This inhibitor also works at the cellular. We treat the MCF7 cell with increasing amount of LATS inhibitor or the negative control. And you can see YAP phosphorylation is decreased. And simultaneously, ER alpha expression is also decreased. Again, it confirms, LATS inhibition results in ER alpha downregulation. And if you look at the YAP target gene, as predicted, they're induced, and the ER alpha itself, as well as the ER alpha target gene, are depressed. And we want to demonstrate that effect, the kinase effect on the estrogen receptor is specific rather than inhibit something else. So we tested the kinase in the YAP test double knockout cells. You can see the inhibitor, LATS inhibitor, reduced ER alpha in wild type cell, but not in the YAP test double knockout cells. And then we look at the effect on cell growth. So in the wild type cells, increasing concentration of the LATS inhibitor, we see nice inhibition of cell growth. In the LATS knockout cell, which are growth slower, and no longer responds to the LATS inhibitor. In the YAP test double knockout cells, also no longer responds to LATS inhibitor. So that's really, and we are pretty comfortable to say this LATS inhibitor, VT02956, inhibit ER positive MCF7 cell growth proliferation is mediated by the Hippo pathway. And we further test that in tumor organoid. So this is the breast tumor organoid, the wild type tumor organoid, and the tumor organoid, we re-express the ER alpha. So this is before the drug treatment, and here after the drug treatment. You can see the ER as a LATS inhibitor selectively inhibit cell growth. Yeah, if we re-express ER alpha, it is resistant. So again, so the conclusion is that LATS inhibitor acts through LATS, or this compound acts through LATS, and ER to inhibit breast cancer growth. Now, For ER-positive breast cancer, there's many drugs you can treat that. However, a significant fraction of them, the patients, will develop drug resistance. And some of the drug resistance, maybe more than 15% of them, are due to mutation in the estrogen receptor, in the ER-alpha. So the ER-alpha is no longer sensitive to inhibitors. For example, the ER-alpha, tyrosine 537 to serine mutation is one of the commonly found resistant mutation. So we made double knock-in cell lines, homozygous knock-in cell lines in the T47D, and then look at the sensitivity to tamoxifen, which is inhibit ER-alpha, or the LATS inhibitor. Nicely, the LATS, these hormone therapy resistant cancer cells are more sensitive to LATS inhibitor than the hormone modulator, the inhibitor. Now, currently, palbociclib, which is CDK4/6 inhibitor, is used in the treating ER-positive breast cancer, and we test the combination of palbociclib with the LATS inhibitor, and you can see palbociclib has the effect. LATS inhibitor together with palbociclib see a very nice synergistic effect. So with that, we like to propose the following model. So perhaps LATS inhibitor such as VT02956 is able to modulate ER-alpha ESR1 gene expression through this mechanism. And because ER-alpha is important for ER-positive breast cancer, and this might be useful for ER hormone therapy resistant cancer, because those that has a mutation in the ER-alpha coding gene are no longer sensitive to like a tamoxifen. So the last a few minutes, I will very briefly talk about another ongoing project is on the semaphorin. Actually, many years ago, my lab has been studied semaphorin exon guidance. So semaphorin initially was discovered as an exon guidance cue. It inhibits exon growth, so it's a repulsive signal. And now we know semaphorin has a very broad biological function involved in angiogenesis as well as immunity. Semaphorin, its a receptor is a plexin. And so we also know semaphorin deletion occurred in some cancers. For example, in lung cancer, there's a reasonable frequency of semaphorin mutation, deletion. So semaphorin came with either secreted, diffusible, or membrane-bound semaphorin that binds to its receptor plexin to signal. The intracellular domain of plexin actually is a gap, a GTPase activated domain, so gap towards RAP. Initially was reported as an RAS gap, which it turned out probably is not true. It's a true gap for RAP1 and RAP2. So Zipperman, a former postdoc in a lab, he discovered that semaphorin induces YAP phosphorylation and inactivation. So here in the wild type cells, if he treated with semaphorin, you can see YAP phosphorylation is induced based on the phosphatase gel, and you can see LATS phosphorylation is also induced. And this effect is completely dependent on the plexin receptor. So he made a plexin ABC, not ABC, A1, A2, A3. So plexin A is the receptor for semaphorin 3A. The triple knockout, you can see this response is gone, and the LATS phosphorylation also is gone. Plexin also, for plexin A to work, it also needs a co-receptor called neuropilin. So we deleted neuropilin, a double knockout, neuropilin 1, 2 double knockout. You can see again, semaphorin, recombinant semaphorin induced YAP phosphorylation is largely gone in the neuropilin knockout, same as LATS. If we look at the localization, semaphorin induced YAP cytoplasmic localization, and in the knockout cells, this effect is gone. So we want to look at if this regulation is dependent on the Hippo. Again, so we test the LATS1-2 double knockout cell. This is a lung cancer cell line, I believe. So, semaphorin-induced YAP phosphorylation, and in the LATS knockout cell, YAP phosphorylation is blunted. And we look at the transcriptome. So semaphorin-induced gene in the wild type and the LATS1-2 double knockout cell. So about a third, slightly over a third of those genes depends on LATS. So I should say the majority of semaphorin regulated genes are independent of the Hippo pathway. But about 36% of the semaphorin regulated gene do require the Hippo signaling. And if we look at the growth, again, so semaphorin will affect the tumor growth. In the LATS knockout cell, this is no longer able to do that. We did the experiment by following, because this tumor cell has deletion in semaphorin, SEMO3B, and we re-express SEMO3B in the wild type cells or the LATS double knockout cell. You can semaphorin re-expression can inhibit tumor growth, but not in the LATS double knockout cell. So Zipeng figured out, elucidated the mechanism. So we know semaphorin binds to its receptor plexin A and the co-receptor neuropilin. And downstream of the receptor, actually the GAP RAP is not involved in the Hippo regulation in response to semaphorin. Although GAP can regulate the Hippo pathway in response to mechanical signal. In this case is RND, another small GTPase, which bound to the membrane, has C-terminal modification stick to the membrane. RND, that GTPase recruit a RAP gap, P190 gap. This gap then results in Rho inhibition. So now we know, we previously very well know, we studied a lot that Rho inhibits the Hippo pathway to regulate gene activity. So again, so this is the mechanism. Zipeng figured that out. So with that, I will stop. I would like to thank the person who did the work. Mainly the estrogen receptor part ongoing project is done by Senghong Mao, and the semaphorin work is done by Zipeng Men. And we get a lot of help from our colleagues and collaborators, specifically Tracy Chen at Vivace Therapeutics, that developed LATS inhibitor and the TAD inhibitor, which is very valuable for our study. And the estrogen receptor, we could not really do the experiment without the advice, invaluable advice from Jeff Rosenfeld. And with that, I would like to stop and thank you for your attention. We'll be happy to answer questions.
Sorry, okay, great. I just unmuted myself. Thank you so much. This is just amazing. As always, your data just very clean, very, all the magnetic details, excellent. I really enjoyed your talk. I think I have a couple of questions here. Okay. This is from Zixing Zhen. Oh, so I might have missed, but does YAP knockout inhibition? Does YAP knockout or inhibition affect ESR expression?
YAP knockout, YAP test, we need to double knockout, slightly elevated estrogen receptor expression. This is not just in breast tissue. We also, I did not show that. We also look at endometrial tissues. We also look at fallopian tube. So yes, YAP knockout results in slightly activation, not a lot, but YAP overexpression is a dramatic reduction of estrogen receptor expression.
Great. Yeah, I remember you have this data in Western, right? Okay, there's another question from Xin Yuan. In the cartoon, you have suggested that the PCP signal modulate Hippo. Can you elaborate on this a bit more detail?
Yeah. So this is none of those work from my lab, mainly from Drosophila, the plano cell polarity. That's like an epithelial, apical basal polarity and a plano cell polarity. Mutation in component of plano cell polarity affects the Hippo pathway. For example, like a disc large, DLG, those kind of things, several of them. I think fat is also maybe involved in plano cell polarity. And as I said, I should emphasize, as I said, the majority are work from Drosophila.
Great, thank you. Actually, Cornell, I have a question. I think this is really interesting because we know, yapatase and KID, that complex on the transcription. Now you found this new one, which is VGL3 and NCOR2. Now this sort of regulates the ESR expression. So my question is actually, have you looked at the patient cohort? The ER positive versus ER negative, and then that VGL3 or NCL, their expression level, how do they correlate?
Yeah. So we did look at that. We analyze analyze the data. So first, the VGL3 expression has very good correlation with the known YAP target gene, CTGF and CYR6.
Right.
So that is one. Now, if we look at the YAP expression in the triple negative breast cancer cell, that's not much correlation. In the ER negative breast cancer, ER positive breast cancer and normal tissue, there's a tendency of decreased YAP expression in the tumor versus the normal tissues. That database, I believe there's way more tumors than the normal tissues, so not everything is matched.
It's very interesting. So they actually VGL3 says it's still required for even the mutant ESR. That's very interesting.
Yeah. So the VGL3 is very interesting. Should really explain now. Tell us TAD can function as either a transcription activator associated with the YAP or a transcription repressor associated with VGL3.
This is very interesting. Yeah, excellent. So there's more questions. So I guess there's no more questions, and then time is perfect. It's almost 5. So let's thank you again. Thank you again, Cornelia. It's really great talk. I really enjoyed it. Okay, so everybody have a nice, have a nice evening. Oh, you there? It's still early. Nice afternoon. Right, it's very nice weather here. I don't know about San Diego. Very nice weather.
Yeah, always.
Yeah, always. We have like over 70 degrees here. Very nice. Thank you so much, Cornell.
You're welcome. Bye-bye.
Okay,
Thanks.
bye.